Welcome to this lecture on developmental biology. Today, we are going to explore a fascinating and critical stage of early life known as cleavage. Imagine trying to share a single massive cake with hundreds of people without baking any more cake. You would have to slice it rapidly into smaller and smaller pieces. This is exactly what happens to a fertilized egg. Understanding how and why these cuts happen is essential because it sets the foundational architecture for the entire future organism. Let us begin by defining what cleavage actually is. Following fertilization, the single-celled zygote faces a massive challenge. It must build a complex, multicellular body very quickly. To do this, it undergoes a series of rapid mitotic cell divisions. However, unlike normal cell division, where cells grow before dividing, the embryonic cell cycle skips the growth phases entirely. This means the total mass and volume of the embryo do not change. Instead, the giant initial cell is simply partitioned into many smaller cells, which we call blastomeres. This rapid slicing is the essence of cleavage. Now that we understand what cleavage is, let us look at the specific geometry of how these cuts are made. To build an embryo correctly, the cells must be cut at precise angles, which we call planes of cleavage. Think of the spherical zygote like a globe of the Earth. A meridional plane slices straight down from the North Pole to the South Pole, passing right through the core. An equatorial plane, on the other hand, slices horizontally across the middle, just like the Earth's equator. Other planes, like sagittal and latitudinal, run parallel to these but off-center. The exact sequence of these geometric cuts determines the three-dimensional shape of the growing embryo. These geometric planes lead us directly to the overall patterns of cleavage, which dictate the final arrangement of the cells. The biggest factor deciding this pattern is the amount of yolk inside the egg. Yolk is dense and packed with nutrients, acting like a physical roadblock that slows or stops the cellular knife. In organisms like humans or sea urchins, where there is very little yolk, the cuts go all the way through the cell. We call this holoblastic, or complete cleavage. But in birds or reptiles, which have massive yolk-heavy eggs, the knife cannot cut all the way through. Only a tiny disc of cytoplasm at the top divides, which is known as meroblastic, or incomplete cleavage. Before we move on to the mathematics, let us see this geometry with a concrete visual example. On the left side of this diagram, you can clearly see the spherical zygote, with dashed lines representing our cleavage planes. The vertical line is the meridional plane, and the horizontal line is the equatorial plane. Now, look at the right side. The top image shows holoblastic cleavage, where the entire sphere has been successfully divided into smaller, equal cells. Below that is meroblastic cleavage. Notice how the massive bottom portion, the yolk, remains completely undivided, while only the small cluster of cells at the very top has undergone cleavage. This visualizes exactly why a bird embryo starts out looking so different from a human embryo. Having visualized the physical cuts, we can now explore how quickly these cells multiply. During this early stage of life, the cells are locked in a highly synchronized rhythm of division. Because every single cell splits into exactly 2 daughter cells during each cycle, the total population does not just add up, it multiplies. This creates a predictable geometric progression, or exponential growth. By understanding this mathematical pattern, embryologists can predict the exact size of the cell population at any given moment, which is a crucial metric for determining if an embryo is developing at a healthy, normal pace. This concept of exponential multiplication gives us a very simple but powerful formula. The equation is n equals 2 raised to the power of n. Let us break down what these symbols mean. The capital N represents the total number of cells, or blastomeres, currently present in the embryo. The lowercase n represents the number of cleavage divisions, or cycles, that have occurred since fertilization. The base number is 2 simply because one cell splits into 2. Physically, this means that as the number of division cycles increases, the total cell count doubles every single time, growing exponentially. A small increase in n leads to a massive explosion in n. Now that we have our formula, let us apply it to a real biological scenario. We are observing a frog zygote in a pond, and we are told it has completed exactly 7 rounds of cleavage. We need to find the total number of cells. We start with our exponential growth formula, n equals 2 to the power of n. We substitute our given value of 7 for n. This means we are multiplying the base number 2 by itself 7 consecutive times. Calculating this out, we get a final answer of 128. Physically, this tells us that after just 7 synchronized divisions, that single massive egg has successfully partitioned itself into an embryo made of 128 individual smaller cells. Our calculation of cell numbers brings us to another critical mathematical concept, the internal balance of these new cells. In a normal, mature cell, the nucleus manages a small, proportionate amount of cytoplasm. However, an unfertilized egg is absolutely massive. Its single nucleus is completely overwhelmed by an enormous ocean of cellular fluid. Because the total volume of the embryo does not change during cleavage, the process of dividing the cell over and over effectively chops that massive ocean of cytoplasm into smaller and smaller packages. Meanwhile, a brand new nucleus is synthesized for every new cell, rapidly restoring the balance between the genetic command center and the fluid it needs to control. To quantify this restoring of balance, we use the nucleocytoplasmic ratio formula. The equation is R equals V sub n divided by V sub c. Here, R stands for the nucleocytoplasmic ratio, which is a dimensionless number. V sub n represents the total volume of the nucleus, and V sub c represents the volume of the cytoplasm inside a single blastomere. Conceptually, this formula tracks how the proportion of nuclear material to cytoplasmic material changes. Because the cell size, V sub c, decreases with every division, while the nucleus size, V sub n, remains relatively constant, the denominator gets smaller. Physically, this causes the overall ratio, R, to increase dramatically over time, bringing the cell back to a healthy, manageable state. Let us see this ratio in action. With a sea urchin embryo, we are given an initial zygote cytoplasm volume of 8,000 cubic micrometers, a constant nuclear volume of 80 cubic micrometers, and 3 rounds of cleavage. We want to find the new ratio. First, we find the total number of cells by calculating 2 to the power of 3, which gives us 8 cells. Next, we divide the total initial cytoplasm, 8,000, by those 8 cells, meaning each new cell has a cytoplasmic volume of 1,000 cubic micrometers. Finally, we plug our nuclear volume of 80 and our new cytoplasmic volume of 1,000 into our ratio formula. Dividing 80 by 1,000 gives a final ratio of 0.08. This proves mathematically how cleavage rapidly increases the ratio from its initial tiny value, restoring biological control. Looking at the result of our previous calculation brings us to a crucial biological realization. Before any divisions occurred, the initial ratio of that sea urchin zygote was a mere 0.01. By undergoing just 3 rounds of cleavage, the ratio jumped significantly to 0.08. This physically demonstrates the entire purpose of this developmental stage. Cleavage rapidly restores the critical balance between the nucleus and the cytoplasm. Without this mathematical restoration, the tiny nucleus would never be able to effectively control the biological activities of the massive cell. It is a perfect example of math driving biology. We have covered a lot of ground today, so let us summarize the most important ideas. First, cleavage is a unique, rapid form of cell division that conserves total volume while transforming a single zygote into a multicellular blastula. Second, the physical geometry of these cuts, the planes and patterns, are heavily dictated by the amount of yolk acting as a physical barrier. Finally, we saw that this process is highly mathematical. The cells multiply exponentially, and in doing so, they successfully restore the critical nucleocytoplasmic ratio, allowing the genetic material to properly control the cell. You now have a solid understanding of the geometric and mathematical precision required to begin building a complex living organism.